Well, my phone says 11:15, so we'll go ahead and get started. My name is Frank López. I'm the Extension Director for North Carolina's Water Resources Research Institute and for Sea Grant as well. And you're in a session today related to water quality and public health. And we have three different speakers from different institutions around the UNC system that are all looking at different aspects of public health as affected by water quality. Just a note for the speakers, at different points in your presentation, I may give you some timings so that we might be able to ask some questions at the end of your session. We'll try to keep things according to the calendar so that people can move in and out of the room and go to other sessions as they prefer. We're going to start off this morning with a remote speaker, Sara Kamanmalek from University of North Carolina, Charlotte, is going to talk to us about doing some geospatial analysis of antibiotic pollution. So Sara, the floor is yours. Thank you for joining us.
Hello, everyone, and thank you for the introduction. Yes, I'm going to talk about geospatial analysis of antibiotic pollution across North Carolina to inform a targeted field study. And first, I will start by providing some background information about the presence of antibiotic and antibiotic resistance in the aquatic environment. Then I'm going to explain the research question of the study, the methodology that we use to answer those research questions, and the key results. And lastly, I will provide an overview of the limitation, conclusion, and dissemination of our results. So there is a growing concern about antibiotic in the environment. There are multiple anthropogenic pathways that can cause antibiotic to end up in surface water. One of these important pathways is wastewater treatment plant treated effluent, which usually contains an elevated level of antibiotic. Another pathway is agricultural runoff from animal farms and manure application as soil fertilizer. As a result, antibiotics have been detected in aquatic environment at concentration of nanogram per liter to low microgram per liter. Antibiotic presence can impose constant selection pressure on bacterial population, which result in the development of antibiotic microbial resistance, which poses human and ecological risk, such as alteration of microbial biodiversity and function. In addition, the World Health Organization has declared that antibiotic resistance is one of the top global public health threats facing humanities. Several states have been reported antibiotic occurrence in wastewater treatment plant effluent and downstream of surface water. These are just a few examples of that. For example, in North Carolina, 10 different antibiotics have been detected in the effluent and downstream of Mallet Creek and Sugar Creek wastewater treatment plant at concentration up to 80 nanogram per liter. So the majority of prior studies have analyzed antibiotic on a side-by-side basis with local knowledge of sampling sites. Here in this study, we focus on the development of a holistic approach to determine streams at higher risk of antibiotic and antibiotic resistance. And we aim to use that modeling results to inform sampling sites. This goal was achieved by a two-step approach. In the first step, we investigated the impact of wastewater treatment plants as a main point source on antibiotic and potential antibiotic resistant occurrence in receiving streams at the national level. In the second step, in addition to wastewater treatment plant, we investigated other antibiotic sources and we integrated our modeling framework to an approach to inform field study quantifying antibiotic and antibiotic resistant genes in North Carolina. In the first step of the project, we aim to answer these research questions. One, what are the predicted antibiotic concentration in wastewater impacted streams across the United States under varying flow condition? And two, how many US receiving streams are at a higher risk of developing antibiotic resistant genes from wastewater treatment plant discharges, and how are at-risk streams geospatially distributed? To answer these research questions, we developed a spatial model that incorporates wastewater treatment plant attributes and hydrology data to predict antibiotic concentrations downstream of wastewater treatment plant. Then we compare our modeled antibiotic concentration to resistant threshold to identify streams with higher potential antibiotic resistant presence across the US. So the first step was data collection. We got geolocated wastewater treatment plant data and attributes from Clean Watershed Needs a Survey and PCS. Then we used NHG Plus version 2 analyzed stream data for hydrologic data. Then we extracted measured environmental concentration and predicted no effect concentration for antibiotic from literature. Our selected antibiotics are ciprofloxacin, erythromycin, and sulfamethoxazole because each represent different classes of antibiotic and they are commonly prescribed as well as they are persistent in surface water environments. We verified the location of 1,300 wastewater treatment plants that are discharging to the stream with a straggler stream order of 1, which are smaller size rivers. And then we inserted shapefiles of wastewater treatment plant and HT flow line into the ArcGIS as a vector layer. And wastewater treatment plant were spatially joined to the closest flow line to locate the discharge sites. We used GIS model to calculate the dilution factor for each receiving stream. And dilution factor is the factor by which the wastewater effluent is diluted in the streams, which is equal to the flow in the stream, which is the sum of wastewater treatment plant effluent and a stream flow of receiving river divided by effluent flow. The safety threshold was set to hazard quotient, and hazard quotients are commonly used to assess the potential ecological risk from individual contaminants. The hazard quotient value for the selected antibiotic is equal to environmental estimated concentration divided by predicted no effect concentration. Environmental estimated concentration is set to median and 90th percentile of measured environmental concentration. And the dilution factor required to meet each hazard quotient is calculated by dividing the hazard quotient for each antibiotic within the treated wastewater effluent by the level of concern, taking into account which was recommended safety factor of 10. It is also important to note that predicted no effect concentration reported for antibiotic do not account for resistant promotion. So we use surrogates for PNEC for resistance selection, which was reported by Beckinson, Palm, and Larson. So here, these figures indicate the map of wastewater treatment plant across the US, color-coded based on dilution factor of the receiving streams under mean annual flow condition in figure A, and 7Q10 low flow condition in figure B. 7Q10 is the average lowest 7-day flow that occurs every 10 years. Dilution factor between 1 and 2 are red dots, and dilution factor above 2 and 10 are yellow dots. For example, the dilution factor of 2 means that effluent will be diluted by receiving stream by the factor of 2, or 50% of the stream are wastewater effluent. As you can see, under low flow, dilution factor drastically decreases in comparison to the mean flow. And our results indicate that dilution factor can be reduced even up to 4-degree magnitude under low flow condition. So because of insufficient stream flow and low buffering capacity of the stream, higher antibiotic concentration is expected downstream of wastewater treatment plant under low flow condition, which will potentially lead to higher antibiotic presence. These are the map of the percentage of site exceeding antibiotic resistant threshold for ciprofloxacin, color-coded, and erythromycin, which is shown as dots, based on 4-digit hydrologic unit, HUC4, and red polygons are HUC4 watershed that more than 80% of the discharge sites within that watershed do not meet the ciprofloxacin resistant safety threshold. As you can see, we only see 11 of those watersheds under mean annual flow condition in figure A, but almost half of US watershed exceeds ciprofloxacin safety threshold under low flow condition. Our results suggest that although sulfamethoxazole concentration was higher than the other 2 antibiotics, the resistant exceedance was negligible due to high PNEC. A straggler stream order defines the size of a stream by hierarchy of tributaries. It has been reported that streams with lower stream order are more sensitive to seasonality and wastewater treatment plant effluent, and that led us to investigate the impact of a straggler stream order on the potential of antibiotic resistant presence. Figure A and B indicate the box and whisker plot of dilution factor classified by a straggler stream order. And the purple line shows the ciprofloxacin safety threshold. 60% of wastewater treatment plant discharge to stream with order 3 or lower, while 93% of a site that exceeded the ciprofloxacin safety threshold are these smaller streams with an order of 3 or lower. Only less than 1% of sites with an order of 6 and higher exceeded the safety threshold. So the main takeaways of this study was that the median concentration for ciprofloxacin, erythromycin, and sulfamethoxazole are 3.4, 1.9, and 6.9 under mean animal flow condition, and 80.8, 44.7, and 161 nanogram per liter under low flow condition. And under average stream flow condition, safety threshold for ciprofloxacin and erythromycin were exceeded in 35 and 2% out of 13,000 sites. But under low flow stream condition, safety threshold for ciprofloxacin and erythromycin were exceeded in 77% and 26% of the sites respectively. So we aim to bridge the national assessment of wastewater treatment plant impacts on antibiotic to the field study in North Carolina. And in doing so, in addition to wastewater treatment plants, now we are looking into the impact of concentrated animal feeding operation, or CAFOs, which is one of the point sources. So this step aimed to answer the question of water concentration of animal.
Sarah, have we lost you?
To answer these questions, we use the multimetric index approach that brings point and non-point sources together. This approach has been developed and used by EPA, and the index will rank most to the least impacted watershed by antibiotic. And that ranking will help us to choose the sampling sites we expect to see the high concentration of antibiotic and potential antibiotic resistant presence. So the index includes antibiotic pollution, which is divided into two categories, non-point and point pollution. Non-point pollution will be CAFOs and antibiotic used by CAFOs per watershed, and point pollution will be the wastewater treatment plant effluent. And then after we calculated the metric value, we calculated the metric score, which is equal to the metric value at a given watershed to the maximum of the metric value. And then we average the score of point source and non-point source to calculate the final score. The index score, as I said, ranked the watershed from most to the least impacted by antibiotic pollution. And then we choose top 3 most impacted watershed. And we locate 3 sites within each watershed to represent the model most impacted sites. And we locate 1 site within each watershed that represents lower human impact as reference sites. Then we collect the samples from these and statistically compare them in impacted sites to the reference sites to see whether sites modeled to be most impacted actually resulted in higher concentration. Here you can see the map of percentile ranking of 2 metrics. To the left-hand side, it is municipal effluent, and to the right-hand side, it is CAFO. Most impacted watershed by municipal wastewater is located in urban area with a large population, while CAFO impacted watersheds are in southeastern and east central of the coastal plain region of North Carolina. Here are top 3 most impacted watershed and their respective selected sampling sites. Orange squares are wastewater treatment plant and pink dots are CAFO. Sugar Creek is urban watershed, and then we have the other 2 watersheds, which are mainly agricultural, and they are located in Duplin County and Stephenson County, which are the first and second largest swine producing county in the United States. This is the antibiotic concentration in the impacted sites and also in the reference sites. And we observed that antibiotic concentration in model impacted sites are significantly higher than reference sites and dominant, and it accounts for 54 of the total antibiotics. And also currently we are analyzing sample for selected antibiotic resistant genes. And these are the average of antibiotic concentration in the impacted sites, and highest antibiotic concentration were observed at the sites that received both from CAFOs and wastewater treatment plant. So this study, especially the modeling part of it, is limited to accuracy and availability of data sets, especially in the state level. We didn't have some data sets that we had at the national level. And some of the industrial sources were not considered because we didn't have any data. And also formed based on a steady state assumption. And we didn't really take it into account the temporal variation. And this study overall showed that we can use the environmental modeling and couple that with the field study to prioritize monitoring and also to use it as informed decision making. And the national study was recently published in Environmental Science and Technology Water, if you guys are interested. And also we're gonna put all of our modeling onto online mapping via ArcGIS, and it'll be available by the end of May. Please email us if you want to have access to it. And I would like to thank our collaborators, especially WRRI, for providing fundings. Thank you.
Thank you very much, Sarah. I do not see any questions in the chat. Is there any questions in the room? If you have a question for her, I'll bring the mic to you so that everyone can hear you, even the people online.
Hi, first, thanks for the presentation. I just have a question. Do you know if there's any work that's being done to look into like potential interaction between the antibiotics that's been discharged and other chemicals like say PFAS or other sort of toxins? Just wondering.
She may be frozen. You may have stumped the panel. I think she's gone. Sarah, did you hear the question?
I did not. I got disconnected.
Let's try again.
Yeah.
Yeah, I was just wondering, like, do you know if there's any work that's being done to look at potential interactions between the antibiotics and the other chemicals that's in the water, like PFAS, which is a emergent contaminant concern because chemical compounds do interact.
I have seen some studies about antibiotic resistant genes and metal, and they're, they kind of escalate the situation, the presence of both of them in the surface water. However, we did not look into other contaminant in this study.
We've got time for one more quick question if anyone in the room has it. I'm sorry. No, that's not. That's me. Yeah. All right, I think we're pretty close to being on time, so I'll go ahead and keep rolling here. Thank you very much, Sarah, for joining us.
Yeah.
Sarah, would you mind hitting stop share on your end, please? That should work. Right is forward, left is back.
So our next presenter is Ryan Paerl of North Carolina State University, and Ryan is going to talk to us about some of his examinations of cyanobacteria.
All right.
Yep. Thank you, Ryan.
Yep. Thanks a lot. Good to see everybody in person. This is a new experience again, having meetings in person. I'm Ryan Paerl. I'm going to talk a bit about probing cyanobacteria for production of taste and odor compounds and cyanotoxin production using metagenomes. So getting right into this, I'm going to start with some background and first cover that the data that I'm going to present today is related to two larger projects where we are focused on, in part, elucidating who the cyanobacterial producers are of taste and odor compounds, specifically in Lake Mickey, which is a drinking water reservoir resource for City of Durham. That funding, that work was in part funded by WRRI and then also the City of Durham. We also had collaborations with Spirogyra Diversified, Linda Ehrlich on that project. And then the second project I'm going to talk about is related to detecting cyanotoxins in the air and water within the Chowan River area. And so that work was spearheaded by collaborators at UNC IMS. And also we received support through NC State's Center for Human Health and the Environment, right? I feel like I'm leaning down. Hopefully you can hear me okay. I'll try not to lean too much more. All right, so general background. We have a lot of modern tools now to actually probe populations that are in situ in environments. And one of the tools that we can use now is metagenomics. What the heck is that? Well, that's also called shotgun sequencing. It's where you take an in situ sample, extract DNA, and look directly at that DNA that's present. You're not going to use PCR, so you bypass any biases that might be associated with that. All right, so it's direct sequencing on what you got in your sample. If you have very abundant things, they're going to show up a lot in your sample. If you have rare things in your sample, you might need to sequence a lot to get at those. All right. There's two common approaches to doing metagenomics, and I'll talk more about them, or we've implemented them in these projects, but the very common one is this one here on the left, where you take these little pieces of DNA, you stitch them together, kind of like a puzzle piece, and make a bigger piece of DNA called a contig. Then you're going to look at those contigs and see if you have your gene of interest, your target of interest there. And in this case, I'm showing GOA. It's a gene related to geosmin synthase, so production of geosmin, a taste and odor compound. We got it there. If you have it, you can then query public databases and see if that sequence matches any known organisms, known sequences that are available. All right. Now, an up and coming approach is the second one, where instead of looking at just those contigs, you take all those pieces, those little pieces of DNA, and you bin them based on their frequency and their nucleotide composition. And you construct what's called a metagenome assembled genome. Getting into jargon here, it's called MAG for short. But the cool thing there is that you not only just look at a single contig, is you have multiple bits of information. You have this genome that's related to a taxa, in this case, Delictospermum that I'm showing here, that has GOA, it makes geosmin, but hey, also it has NIFH, which is something that encodes for nitrogen fixation. So then you can say, hey, there's a link here between geosmin production and nitrogen fixation. Hope everybody's still with me. All right, so getting into the projects here. The first one I promised to talk about is Taste and Odor project. We were focusing on Lake Mickey, okay? This is one of two major drinking water reservoirs that supports the drinking water needs of City of Durham. The City of Durham Water Management group there gets annual complaints about taste and odor issues, especially in the spring and summer. So it's of concern to them what is actually a source of these taste and odor compounds that their customers are detecting and uneasy about, concerned about. We don't know if it's cyanobacteria. We're starting off here. We didn't know if it's cyanobacteria, maybe other bacteria. And the lack of knowledge, arguably, I'll argue, limits our ability to actually predict when these peaks in T&O concentrations happen, and also possibly taking mitigation tactics to reduce that from actually being produced. You might want to use certain nutrient reductions to reduce the success of nitrogen fixing populations, for example, or different heterotrophic bacteria. So knowing more about the sources could be helpful on that front. All right, what we actually did, we did some time series sampling in 2020. We collected weekly measurements actually from April, or weekly samples, excuse me, from about April to October. And we also did this specific serial filtration for metagenomics. So we're basically trying to enrich for large versus small organisms, aka large cyanobacteria. We could get more information on them by physically separating them out from all the other bacteria there. We also had, with our collaborators, City of Durham Water Management, and also Aspire Diversified, we have other really valuable metadata that go along with our sampling for metagenomics, especially measuring these two compounds here, geosmin and 2-MIB for short. So City of Durham Water Management covered us on that front, but we were able to therefore pick some samples that were particularly rich in these T and O compounds and use our resources wisely for sequencing for metagenomics. All right. So some results on this front. We focused in on 20 different samples to actually sequence. I can tell you more about sequencing if you want. I'm just going to bypass that right now. But we actually sequenced about 500 billion nucleotide bases, so a huge amount of DNA information there. When we looked at those contigs that I talked about before, those long pieces of DNA, and actually searched for some of the gene targets that encode for production of these TNO compounds, we got 16 hits amongst all of that data. Most of those hits to these genes were in the larger size fraction, indicating that they're either filamentous, large organisms, or they're particle associated. And specifically, they were to these organisms. So we had a high number of hits to Delictospermum for GOA. That produces that geosmin compound that I briefly talked about. Also an unknown, unidentified, uncultured bacterium. A distant hit to Oscillatoria, another filamentous cyanobacteria. And then we only had one hit to the gene encoding for 2-MIB, the other compound that I mentioned. And that was distantly related to microcoleus, which is pictured here. We're not 100% certain that it is microcoleus, to be determined on that front. All right, so using that more modern MAG generation technique that I was talking about, these metagenome assembled genomes, we were able to construct about 271 of high quality MAGs from that data set. Among the cyanobacterial MAGs, the Delictospermum ones that we obtained all had GOA, so indicating these are probably really key producers of geosmin. I'll add with these MAGs, these represent abundant populations. You're not going to get any MAGs from more rare populations. All right, so Delictospermum is abundant. It also has GOA. Looks like it's a good indication that it's a geosmin source. We surprisingly detected actually that Achloroflexi had the ability to make geosmin, had GOA genes present in them, and that's not evident in the literature. Ecologically, that's a head scratcher. Why the heck are they doing that? And we also did not detect any of those 2-MIB genes in these abundant MAGs. So meaning they're rare populations that are likely producing that 2-MIB compound. Just very quickly, I wanted to show you this computational tool that we're actually using to look for these genes and the ability to make secondary metabolites like taste and odor compounds. It's called anti-smash. I can talk with you more about it if you want, but using this tool, you can actually see a number of other compounds that these organisms, these MAGs, basically have the ability to produce. All right. A quick shout out as well. We saw to work from my PhD student, Hua Wang, who's also going to present more data on this taste and odor project in the poster session. Concordant or in agreement with that MAG information, we could see that also Delictospermum biovolume and concentration of geosmin had a nice positive correlation. So giving us more confidence that, okay, we're feeling good about that genetic data. We have confirmation on the chemical and also microscopic side that it looks legit. They're strong sources of geosmin. For those of you that are up on filamentous cyanobacteria and taste and odor production in the audience, you might ask this question, well, didn't we already know that Delictospermum can make geosmin? Well, not exactly. Turns out a lot of different strains, if you look at them, I'm just showing you a diagram here of genomes from different Delictospermum strains, have different capabilities. So it's not surefire that you're gonna see geosmin production if you detect Delictospermum in the environment. Okay, it gets really complicated. There's different varieties that make cyanotoxins. There's some that make taste and odor compounds. You can't just predict that based on detection of Delictospermum. All right. So quick take homes here. We think that Delictospermum is a very important geosmin producer and probably a problematic species or genera, I should say, in the future at Lake Mickey, because it fixes nitrogen. Conditions are going to get warmer also in the Piedmont of North Carolina. That favors nitrogen fixers on that regard. So something to keep a lookout in the future. We had difficulty clearly detecting the 2-MIB producer. And the last thing is, amongst these cyanobacteria, we actually couldn't find any evidence that small picocyanobacteria that are really abundant actually in our data and abundant in the field. We couldn't find any data suggesting that they were actually taste and odor producers. So it's really all about these filamentous cyanobacteria. Okay. All right, I'm going to go quickly. I'm running long. The second project, we are focusing on the Chowan River. This is an area in the northeastern North Carolina that's known to see blooms of cyanobacteria in the summer and fall. Some colleagues also at NC State amongst NCDEQ folks have shown that there can be very rich hotspots of cyanobacterial blooms in certain areas. Microcystin concentrations can be really high. And it still remains unclear who are actually the producers of microcystin because we're lacking direct evidence there. So going after that question, we also did some time series sampling from two sites within the Chowan River in collaboration with folks at UNC Chapel Hill. I'm short on time. I won't go in depth on this, but similar story. We collected similar samples, got good metadata, had a key collaboration with folks at the CEEG, which gave us access to these sites. It wouldn't have happened without them. Big take home here. We really only sampled and observed one major bloom during 2020, and it was in July. We could not detect microcystin in the air, well, to high levels. It was very low where we could detect it. And we were asking, why can we not detect it in the water or in the air? Turns out when you look for those microcystin genes, they're hard to find. And of those genes that we could actually find, Microcystis was the genera that had them. They were just rare. Okay, we generated mags as well in this study. The most abundant ones were Delictospermum and Abena-like. They lacked microcystin genes. And so an emerging story that we're seeing here is that actually looking into those mags, we don't see kind of the big three, I would argue, of cyanotoxin production, the genetic evidence of those three common cyanotoxins, microcystin, saxitoxin, anatoxin. We don't see any evidence there. Instead, we see a lot of these other understudied compounds, genes related to their production being present in these bloom forming organisms, these Delictospermum, really common filamentous cyanobacteria. These aren't currently measured. So we think that on the horizon, it's worthwhile to spend a little more time or future attention into actually trying to quantify them, study them a bit further. All right, take homes here. We saw ephemeral blooms. We think that, but we could not find any microcystin production linked to those bloom formers. We think these other secondary metabolites might be really important to study in the future. People have shown that they do have toxic effects on nematodes, fish, actually, and are concerned for ecosystem health, possibly human health. All right. Broad take home here. I think we have found that metagenomics offers really valuable insight in what the capabilities are of in situ populations. It's tough for us to extrapolate just from observing Delictospermum being present there, what is actually possibly going on with metabolism or production of compounds. I'll leave my acknowledgements up here because I've already gone long. Thanks for your time and attention.
Thank you, Ryan. Actually, you're fine on time. So we actually have some time here for a question or two. I don't see anything online. Is there anything in the room? Dr. Fear.
Hey, Ryan, that was really cool data. Just curious, could you take it to the next step? How does the city knowing what's there help them better manage it?
Yeah, very good question. So, well, one thing that is of use, I think, is on the side of well, considering management of certain nutrients, especially phosphorus in the case of Delictospermum being such a clear geosmin producer, it's going to do well when there's plenty of phosphorus, other nutrients around, so that it could compete well under low nitrogen concentrations, conduct nitrogen fixation. So it may give them more incentive to consider how to deal with legacy phosphorus. I know that's a messy thing. Dredging up phosphorus is not cheap. And also, yeah, just a difficult task, a big engineering task. This data also can be used to help monitor. So if you wanted to detect maybe in real time quickly that, hey, we have a population that could be problematic, you may be able to do treatment in a rapid succession to try to cope with spikes before they actually get too high and make it to customers. You should check out WRRI's poster. In the afternoon, we actually looked at some treatment techniques using earth tech. It's a copper sulfate algicide. So that's something that has been used. You may or may not want to use that based on some of the data we presented in terms of other secondary metabolites being produced. That's a little bit further than your question, but is that helpful?
Undergoing a project. Oh, thank you. My understanding is the city is undergoing a project to install aeration in Lake Mickey. Do you know any more about that? That probably will have an effect, a beneficial effect.
I don't. Yeah, that's a really good point, and I didn't think of that on the fly here. But that, yeah. So nitrogen fixers definitely might be impacted by that. Less stability in the water column. They are very buoyant too. So there's the possibility that they won't be phased by that mixing and resist it. But that's another thing to consider. It might help guide some decision making there. Yeah, definitely.
One question. I know with the first study, you were running some lab tests to get concentrations of some chemicals of comparison, but I didn't see, I may have missed it, but I don't know if you had any lab analysis done in your second study for microsystem to compare with the metagenomic data.
Yeah, I went very quickly. We have particulate and dissolved measurements that were done by GCMS, mass spec based measurements, and all of those basic that we collected showed that the levels were quite low in the nanograms per liter, tens of nanograms per liter, and then many were undetectable. So yeah, good question. Any others? Nope. Oh.
So you mentioned that with this method, you miss out on some of the rarer species and that you didn't see anything for the 2 MIB that could be producing that. So I was just wondering, like, what potential impact, what's the likelihood that some of those more rare species could be impacting these blooms?
Yep, it's a constant problem to be aware of, for sure, whether or not rare populations could have some ecosystem level impact. that's a limitation to some of this. It's also a limitation for PCR. That might be one way to try to tackle that, is you say use a PCR assay to try to detect them. But those are biased too, because you have to know what your target is. In general, the 2-MIB issue that you mentioned is interesting, and maybe it is a really potent producer since it's rare, but we can't fully answer it. Good question.
Thank you very much, Ryan.
Thanks a lot.